Hi everyone. We're going to start the session. I'm Joey Belcher. I'm on the board of directors here for AMCSI. I'm the secretary. Thank you all for coming. I'm excited to have our presenter, who is a professor at Drexel University in the pediatrics department. Went to Stony Brook for many of his degrees, including his MD. First time at conference, so make sure you give him a warm welcome. But thank you for being here, Doctor. We really appreciate it. Thank you.
Good afternoon. Can everybody hear me?
Yes.
Good. Well, I guess I'd like to start with a question. How many people have been to a geneticist or genetic? And maybe about half or 60%. How about genetic counselor? Both. So what I will talk about may be somewhat new to some people and somewhat of a reinforcement, but as Dr. Hall said, genetics is always changing, and hopefully I can add things that she hasn't touched on, or touch on things that she's discussed in a little more depth. But my goal is to discuss the genetic evaluation process with a focus on genetic testing. We'll define key terminology: chromosomes, copy number variation, DNA sequence, next generation platforms, whole exome sequencing. These are sort of like a hodgepodge of different types of tests, and they do get confusing, and they do sometimes get overwhelming to families when they're presented with these options to help make a diagnosis as well. And we'll also discuss the process of genetic counseling. So, as Dr. Hall mentioned in her opening lecture, just not even an hour ago, human beings are about 99.9% or so identical at the DNA level to one another, which is quite a lot. And that's somewhat of an astonishment when we see that our population is made up of many faces, which define our population. Individuals are different from one another, and we, as geneticists, we celebrate those differences as well. And individuals vary in genes that they possess, which is called genotype, and the physical characteristics they acquire as a result of an interaction between genetics and our environment as well. So there's the genotype, and then there's the phenotype, the outward manifestation, what we see outwardly that makes us appear to be different from one another. So Dr. Hall had enumerated on the reasons for a genetic evaluation, and these include obtaining a more precise diagnosis, offering genetic testing, enable a better idea of natural history, treatment options, information about risk for future pregnancies, and helping a family work through their own ideas about how their child's condition may have happened, and helping to address parental misconceptions and alleviation of parental guilt. All of these are important. So I'm going to discuss what is the genetic evaluation process and how does that take place, what are the components of it as well. And some of the key people include a genetic counselor who is a master's board certified in the United States. I'm not sure if everyone here is in the United States or from other countries, or but there's an equivalent depending on where you are in terms of board certification and professional licensure and recognition, depending upon where you live. Clinical geneticists are typically MD and board certified, and genetic counselors can work by themselves. They can work in conjunction with clinical geneticists, but they can also provide counseling by themselves. And where I work with two Stanford genetic counselors, and we work together, but they can also work by themselves and provide information and resources for families. So the genetic counseling, the genetic evaluation process starts with a history, as any other medical history goes, family history, physical exam, and other ancillary information, medical tests, x-rays, genetic testing, genetic counseling, and addressing psychosocial issues. Sometimes it's the psychosocial issues that actually are mentioned at the forefront, and that may be number one of what's on the patient's mind. They may say, well, why did this happen? Or I feel like I may have caused this problem, or it may have been something that I did. So that may be the number one reason in the family member's mind. And it's the genetic counselor who will contract with the family and will identify what is the reason for them being here or their child being here. And that's a discussion that we have at the beginning of each session to find out what's really in the mind of the patient and the family, and why are they here. It might not necessarily be the reason that the provider referred. So I'm going to target a lot of this towards arthrogryposis, and a lot of this has been identified by Dr. Judy Hall and colleagues in other publications that I have cited at the bottom of my slide. So if we're talking about arthrogryposis, we really want to get a good prenatal history, and we want to ask about maternal illnesses, a fever, greater than 39 degrees centigrade, injury, medications, drugs, and alcohol. These are components that need to be addressed, and Dr. Hall had identified these in other research studies that she's published and worked with colleagues. And she's, as you all know, the most knowledgeable clinician regarding the genetics of arthrogryposis and environmental causes as well. So a lot of what I know about arthrogryposis I learned through her writings. Also, it's important to address maternal bleeding, uterine anomalies, history of threatened abortion. Did the family have ultrasound? Did mom have ultrasound? Was there fetal growth that was observed? Were there skeleton or other major organ types of anomalies as well? Remember, Dr. Hall mentioned a one-third isolated limb, one-third limb plus other involvement, and one-third limb plus central nervous system involvement. And that becomes important as geneticists when we try to think of what caused the child's arthrogryposis. Did the family have any prenatal studies as well? Amniocentesis, non-invasive prenatal testing, chromosome microarray. And I'll explain what a chromosome microarray is. How was the weight gain during pregnancy? Was there excessive weight gain, which could have been due to fluid polyhydramnios, or was there decreased amount of fluid or oligohydramnios? How was the delivery? Was it vaginal? Was it cesarean section? What were the parameters at birth? So all of these are part of the history that should be part and parcel of most medical types of histories. And then after birth, what about hospitalizations, neonatal course? Were there any types of surgical procedures, which we know for arthrogryposis there are certainly many types of surgical procedures that are involved. And a developmental history with attention to motor milestones, speech, personal, social types of skills as well, and review of system which identifies other types of malformations and different types of organ systems. Family history should center around degree of relatedness between mom and dad. For instance, consanguinity means common ancestor, and individuals who are more closely related to one another share a greater number of genes in common than unrelated individuals. Were there miscarriages, stillbirths, parental heights are important because many arthrogryposis-related conditions can be associated with short stature as well. And if we're looking at or suspect amyoplasia, one would expect to see an absence of family history of contractures. Dr. Hall showed many slides in the last talk, and many of you are familiar with what the physical features associated with different types of arthrogryposis are. The physical exam is typically done by a medical geneticist, and for those of you who have, or their child who have had physical exams, most medical geneticists will do many measurements. They'll come in with a measuring tape, measure ears, eye distances, filter chest circumference, hands, foot, and body proportions as well. So we do a lot of measurements. It's not because we are singling anything out. It's part of what we do. It's part of the medical genetic evaluation. And all of my patients pretty much get the same set of measurements. Some may have body proportions, and others who have normal stature probably won't have body proportions as well. But by and large, most patients have the same set of measurements, the patients that I see. And then we do a head-to-toe evaluation. We look for eye spacing, facial contour, chin being small or large, palate being high-arched, clefting, limb creases, palmar creases, hands lengths, finger lengths. And then obviously we pay attention to the joints, whether or not there are congenital contractures, where and which joints are the contractures, are they symmetrical as well? Are there dimples over affected joints, foot deformities, birthmarks, such as a nevus flammus over the forehead? And we'll also look at radiographic information, such as long bones, which might appear to be gracile and osteoporotic. All this information, a lot of the ancillary information we can review from records that are handed to us. It's really helpful if you're seeing a medical geneticist to get records to him or her in their clinic in advance of the genetic evaluation, because we, like pretty much most centers, we like to review our cases the week before. So we have a weekly planning meeting where we can sit down and go over things, and I look to see what x-rays do I need to look at ahead of time, and what conditions do I need to read up on ahead of time, and things like that. So we go into the session with some degree of information so we can help the family better address their needs and their concerns as well. So a lot of what geneticists do and a lot of what we see involves pattern recognition. So Dr. Hall mentioned about fetal akinesia, and this, what we try to do is look for components, and we look for key or cardinal features, and physical exam can help tell us information. We look for, in this slide, wide-spaced eyes, which also means hypertelorism, ears that can be low-set, contractures of the fingers, foot deformities, small chin, and this set of features, where there's also underdevelopment of the lungs, goes along with fetal akinesia. So this is a set of features that are all clinically related to one another, and that we can look at a pattern, and we can say that the history and the set of physical findings are consistent with fetal akinesia. Now, we can go steps further and say, is there a gene, or what genes would be thinking about? But we look and we look for patterns, and then that helps us localize a particular subset of conditions that can be associated with arthrogryposis. So then we can say, okay, what's in the literature about this? What kinds of tests can we look at that can help provide information for the family? So I know a lot of families are new, and genetic testing is always changing, so I want to spend a little time about testing, because that can be something that is challenging to really grapple with. So I'm going to spend a little time just on genetic terminology. As Dr. Hall mentioned, we have 22,000 hereditary units or genes that are distributed on 46 chromosomes, 23 pairs. So we start off with a level of organization with the chromosomes. We can envision chromosomes as a set of encyclopedia books, and we have 46 encyclopedias or chromosomes that are in every cell in our body. And chromosomes then contain the genes, so that's the genetic unit, and genes are made up of DNA. And we can have genetic mutations, which are alterations in what we call the spelling of our genetic material. So here is a nice slide that is actually from the NIH, and it really helps to discuss the level of organization. We start off with our genome, and then chromosomes. So remember chromosomes? We talked about the encyclopedia. So in a set of encyclopedias, you can envision that our genome has 46 encyclopedias or 46 chromosomes here. And next to the encyclopedia are the chromosomes, 23 pairs. If we're a male, we have an X and a Y chromosome. If we're a female, we have 2 X chromosomes. And they're laid out like this. So if we count, and I counted the number of encyclopedias here, there are 46 here in this set of encyclopedias, so 46 volumes, and each volume is a chromosome here. So when you would order a chromosome study, we would look to see are the volumes correct, are the number of volumes correct in our set of encyclopedias. Then within each volume, there are chapters and pages, and that's another level down, and we'll talk about chromosome microarray. But chromosome microarray will enable us to check to see if the pages are properly there or if the pages are not there. So that's then a level of organization at a higher level of resolution from the chromosomes. So we're talking about, we have volumes, now we have pages, and then now we can go a level down further, and we can look here with the magnifying glass, and we can look at specific sentences, and then we can go to the level of the gene and the level of the DNA. So it's all about levels of organization, and that's what genetic testing is all about, the level of organization, and how as a geneticist will think, I'll think mechanistically, do I think chromosome abnormalities have been ruled out adequately? Do we need to do a special test for that called chromosome microarray? If I don't feel that's an issue, then I'm going to go a level then higher, and I'm going to think about the level of the gene, and I'm going to think about DNA sequence approaches, which can be single gene, or we can do panels, or we can go big guns and do whole exome. And I'll talk about all of these in depth. So when I started my career as a clinical geneticist, we were in this era, we were somewhere in between whole chromosomes and chromosomal banding. I started my clinical training as a geneticist in the late 80s. So at that point, we were able to do what's called a karyotype. That's an ordered chromosome array, as I mentioned. And people were able to stretch the chromosomes out and do really what's called high resolution banding towards the late 80s, and look to see if there's extra or missing material that would have otherwise been missed if you didn't really stretch out the chromosomes. So in these situations, we're looking at an order of close to 100 million or so in terms of our DNA bases, which make up genes. And then after high resolution banding, we went into the era, this is somewhere in the late 90s, early 2000s, to comparative genomic hybridization and fluorescence in situ hybridization chromosome microarrays. Now, and pretty much a chromosome microarray is where a lot of the frontline testing starts to see if there's copy number variation, meaning too much or too little of a chromosome segment. And now we are actually in the era of whole exome and whole genome sequencing, where we can get down to the level of one nucleotide or one letter in the alphabet. So We'll talk a little about chromosome microarray. Why do we perform chromosome microarray? When I started genetics, we did chromosomes, we didn't do microarrays, so we were only looking at books or volumes of the encyclopedia. We were not looking at pages. But back maybe about 15 years or so, we began to then look at pages. So really a higher resolution in terms of what we can see. And the reasons for doing this are still the same. If we see a child with multiple birth defects or even a single birth defect, if a child has cognitive impairment, so as Dr. Hall mentioned, arthrogryposis with central nervous system involvement, that would be a reason for getting a chromosome microarray. Autism, facial dysmorphology, or changes in facial features which appear to be distinct and part of the syndrome, short stature, failure to thrive, unusual ultrasound findings, or parents may have what's called a balanced chromosome rearrangement that was previously diagnosed, and their child will be at risk for unbalanced chromosome changes, or abnormal prenatal free fetal DNA findings. So free fetal DNA, you can say that 10 times fast without the sidebar times saying it fast, but it's a blood test, and that enables us to be able to see if there is evidence for aneuploidy such as Down syndrome, trisomy 18, trisomy 13 in the fetus. That needs to be confirmed on subsequent testing, either after birth or amniocentesis, following genetic counseling as well. So this is what a chromosome microarray study looks like. You always need, at this point, a blood sample, and we are always comparing the patient's DNA with a control DNA from a copy-neutral number individual as a control. And it's amazing that you can isolate the DNA, and then you can put the DNA on this chip that has like several thousand wells that you really can't see with the naked eye. And each of these wells contains what we call a genomic region, so a region on our genome. And the one by one, you are looking to see if your patient's DNA is present in the right amount, or if it's present in less amount than it should be, or more amount than it should be compared to a control DNA. So you have to have your patient DNA bind with the DNA in each well, and then you are looking to see if equal or if there's dosage loss or dosage gain. So you can see if green is patient and orange is the control, you want to see an equal number or an equal ratio of control in patient DNA for each well, by well, by well, by well, by well. So you're going, thousands of wells during this whole process, and you have a machine that is keeping track of whether or not there's copy variation. So in here you have equal hybridization, so equal amounts of your patient and your control DNA. But here is a situation here where you have dosage loss. So you have, relatively speaking, 3 of your orange lollipops compared to 1 of your green lollipops here. So for that particular region, there is a decrease in copy variation, copy number. And here you have dosage gain. So for each orange color in this region, you have 3 or copy number excess for that particular region. So this is really just doing a determination to see if each region is present in too much, too little, or the same amount. And the computer will read each region. This could be, the whole genome. And here, everything looks fine. This is your baseline, or 0, meaning no difference between your copy number amount for that particular region. But when you get here, and you can look, and this is a dip down over a large region. So it's consistent across this whole region. And here's the chromosome here. So for this particular region, this is the short arm and this is the long arm. This is chromosome number 7. There is a consistent loss over this tiny band on chromosome number 7. So this individual then has a what we call a deletion, copy number loss in this particular region. So not all copy number gains or losses can cause disease. The lab reports this, and then they have databases that they look at, and they say, well, is this significant or not? And whether or not it's significant relates to the size, it relates to what genes are in this area, it relates to what papers have been published with similar changes in this area. So people have their homework to do, and when we get reports, a good side of genetics lab will have gone to the databases and provided us with some information and guidance upon how to interpret this result, and then we'll review what they find, and we can also, we have special databases that we can look at and see what particular genes are present in this area as well, and we can then provide information to the family as whether or not this is significant, insignificant, or we're not certain, and we need to continue to follow from time to time, because sometimes over time, as Dr. Hall mentioned, genetics changes over time, so going back to a geneticist every few years to have to check in and see if there's any changes in the information is always helpful. So again, just showing you here, you have a test and a reference DNA, and we are looking to see if either there is gain or loss in the test DNA compared to a control, and we can identify whether or not it's copy number gain, loss, whether it's significant clinically or not significant clinically, and then provide that information to the family. Here is a side-by-side picture of a chromosome. We call it a G-banded karyotype, and this is what a chromosome microarray looks like. This is each chromosome, 1 to here, 1 to 22, and 23 is the X chromosome here. So also a chromosome microarray lets us know if the information is there or not. It doesn't tell us if the information is rearranged differently than it should. A karyotype will give us that information. So you can have the proper amount of genetic material, but you can have a exchange of material, say from chromosome 21 to chromosome number 14. So that's one type of rearrangement. There are an infinite number of chromosome rearrangements, and as long as the chromosome amount is the way it should be, a chromosome microarray will not tell us anything that is unusual. So let's then go to another level of resolution. Let's talk about the gene. Genes are complex, and when we want to do gene tests, for instance, say the child has had a chromosome microarray and that's normal, but the geneticist still suspects that something else is going on. There are other types of genetic and genetic sequencing tests that could be altered potentially. And here is a diagram of what a gene looks like. In order for genes to be made, there's a turning on and turning off switch called a promoter. It's kind of like a regulator or a rheostat. So you have to turn it on here, and this will allow the gene to be expressed. And genes are made up of what we call exons. So these exons could join together to make the gene that is then going to be made into protein, but there's introns that have to be taken out. So genes are made in pieces. So here is one piece, here's another, here's another, and this is just the orientation. 5' is the beginning, 3' is the end of that gene. We have a start region and a stop region here. And these regions are just taken out. And you have joining of what's called the exomes. And you have taking out or splicing out of the introns. There is a code for, it's called the genetic code, and genes are made up of letters in the alphabet, and these are called nucleotides. 3 nucleotides then code for what's called an amino acid. The amino acids are joined together to form a protein. So you have, this is proline, this is arginine, and you have starting codons and you have stopping codons. But you can identify what a gene does by backtracking and looking at the sequence of the gene and then figuring out what protein should be made. Sometimes there are spelling errors in that particular gene, so you can have a change here. And some changes are neutral, some changes affect the protein structure, some changes cause the protein not to be made fully, and it stops earlier than it should. So that's important for us to know about and for us to understand if we're trying to understand the genetic reasons for having a genetic condition. We can sequence individual genes, and that the name Sanger is the name of a famous biochemist who developed a sequence technology called Sanger sequencing, which I'm not going to go into, but in principle, it's spelling letter by letter by letter of a particular gene. So it allows us to then, you have your particular DNA, and using the DNA and special chemical reactions, we're able to know what the letter is for each and every part of that gene. Is it the right spelling? Is there a problem? Is there a spelling error? So this is Sanger. It's a single gene sequence analysis. So this is a technology that was being used 20 years ago. As well. We can only sequence one gene at a time, but as you heard from Dr. Hall, we have 22,000 genes. And if we wanted to sequence all of them by Sanger sequencing, we would be here for quite a long time to do that. So it's now with the advent of what we call high throughput, meaning many genes all at one time, being able to be sequenced at once, we can do a lot, and we can look at those pathways that Dr. Hall mentioned more effectively through the use of next generation sequence analysis approaches. So another important feature of DNA sequencing is looking at the reading frame. So we can figure out the spelling of a gene, but we need to annotate it properly. We need to know where it starts and where it begins. Otherwise, it's just going to be somewhat gibberish. Now, I'm sure people who are astute can see some words in here. Somewhere there is the word quick. Somewhere there's the word fox. So we want to know out of this alphabet soup, what is the important part, what is the part that's really coding for a particular protein, and you have to make sure you're reading the gene properly and you're starting at the right starting point. Because, as I mentioned, letters, groups of 3 are important because it's 3 base pairs that then code for one particular amino acid. We want to make sure we're in the right frame. So now if I were to highlight also all the important parts, you can see the quick brown fox jumped, so the jump is here, over the lazy dog, the dog lay quietly dreaming of dinner. So this is what this looks like when you get rid of all the introns. or the non-coding regions, and then you highlight what is important. And this is what we have to do when we sequence a gene, when we analyze it as well. So we want to get rid of information that is important, but it's not important in us understanding, in most instances, the nature of the genetic effect. There certainly are changes that can occur here in the intronic regions that may bear on how genes are put together in that. But a lot of the bulk of the important features are in these exons. So when we talk about exomes, and I'm sure everyone here has heard of exomes, an exome is the sum of all of the coding regions of our 22,000 genes. So one exome is going to equal all of the exons of our 22,000 genes. So here is, again, one gene with introns and exons, and we're looking at all of these exons from all of our 22,000 genes. So if you look at a whole apple, meaning the whole genome, so the big apple, and the exome is 1% of that. So it's really small, and we can offer whole exome testing in the clinic, but you should also realize that we're only looking at 1% of the genome, and as Dr. Hall mentioned, a lot of the genetic material at this point is regulatory in nature. We don't really understand what it does, but this is information that is important and that we could really look at as well. So when we talked about alterations for chromosome microarray for exome analysis, the types of findings we can get are very similar. Every person will have variations in their genetic material that make him or her unique. And most of these changes are not going to affect our appearance or risk for disease or behavior. In the lab, there are 4 types of changes that we may find in our genome. And these can be normal in nature, which are not disease-causing. These can be disease-causing as well. These may be disease risk types of association, which not necessarily may cause you to have a particular disease, but perhaps in the exposure to other environmental agents, this can increase your risk for having a particular condition. And then there's this gray area of unknown or variations where we just do not know whether or not they're clinically significant at the present time or not. And we call those of uncertain variations of uncertain significance. So this is a large pedigree, and as a research tool, whole exome came out first, so that studies like this to understand or underpin the inheritance of large families or even small families where we felt the likelihood of identifying the genetic cause would be great. And it would be hard for us to go. Imagine Dr. Hall mentioning over 400 genes, and if we had to sequence each of the 400 genes at a time, and we had to send you back, a family back to the clinic and have 400 separate blood draws, and there's 400 tests really to do that. Well, now with the advent of next generation platform testing, we can do a lot all at once. So here, many people with arthrogryposis, it's a neuropathic type. There are a lot of double bars here, which means that there are common ancestries. Parents are consanguineous, meaning that they are related to one another as well. And using, this group didn't look at every single member in this family, but they were able probably to do this study and use a few key members in the family and their parents as well to identify a set of genes that they felt would likely be candidates and could cause this particular phenotype. So we started with Mendelian genetics, and that certainly advanced into the era of human genetics as well. And So the principles of Mendelism have really been applied and carried over a lot to form human genetics, which is what I do. We could use this to better understand diseases and disease processes. So this is a picture of the patients in this family, of some of the patients in this family, and you can see the contractures that are evident. The exome analysis identified ERG1C1 from what we call whole exome. I'll go into that in a minute. But you can see the power of this technique, which was used originally as a research test to identify genes that could be at play in conditions where we thought there would be a high likelihood of a genetic cause. So there are different types of DNA analysis. We talked about Sanger. Now we'll talk about platform types of DNA analysis and whole exomes. And these all go under the umbrella of next generation. So this is now what's really used quite a lot in the clinic when you go see a geneticist. We certainly avail ourselves of what we can offer families to help them and their doctors find the cause. But again, we're looking at analysis of billions of DNA strands in parallel all at once. It's pretty mind-boggling. Much more high throughput than a Sanger sequencing, which you can do one at a time, and technically it's a lot easier than Sanger as well in terms of how one can do this. So this is a little techie slide, but I just want you to get the concept that we start off with. This is everything in our genome, genomic DNA, and we fragment it. Then we select for regions that we know we can sequence. I mean, this has been looked at in other types of studies, but we are finding regions that are representative of all of our coding genetic material. We capture the DNA, and then we are able to sequence that, we're able to align it to a reference sequence, and we're able to identify variants. So what we're doing is looking at everything, and we are sequencing all of the 22,000 genes in principle. Not everything gets sequenced equally, so it's up to the clinical geneticist. The analysis has a lot of what the clinical geneticist sees when the genetic counsel reports to the lab as the phenotype is very important in terms of how the lab does the analysis, because they will get variants and they want to know, well, what does this child have? They want to know, does this child have congenital contractures? Does this child have central nervous system problems? Does this child have a heart problem? What is the craniofacial phenotype of the patient? Are the eyes wide spaced? Are the ears low set? So they want all this information, and they look through bioinformatics. They will look at all the genes that come up as interesting in terms of sequence variants, and they'll say, oh, we have a gene X, and this gene X is expressed in muscle cells, and that could be very important if the child has arthrolycosis. So they will really need to know the phenotype, and they like our reports. They want us to fill out checklists of system by system in each organ of what the patient has, and that's very important as well. So geneticists and genetic counselor are trained to do that. And we then review the variants. So this is just a slide showing you that we start, the lab will start with a referral for exome and genome sequencing as well, and there will be, this is the process. We start off with a referral for genetics, genetic counseling, genetic evaluation, and then there will be some preliminary results. The genome team, people in the lab review this. In some instances, often we're talking about several months to get a result back for whole exome sequencing analysis as well. So there's a lot of, the sequence part, you're running the DNA through a machine, and that isn't taking too long, but it's the analysis, the downstream analysis, what we call bioinformatics, having the laboratory and a lot of people look at the variants and say, okay, is this present in the parent as well as the child, are the parents affected or not? If we see a variant in a child and there's unaffected parent, it may be less likely to be significant than if we just see a variant in the child and we don't see it in their mom or dad who are clinically unaffected. So all that information needs to be presented to the laboratory. And then the lab will hand us a list, a report with, maybe they'll say this is the one that's likely to be disease-causing, or they'll give us several, and then we have to go to the literature and review papers, pull out papers, and go to our references and say what it is we think. And sometimes we're not really giving the patient a definite answer, and sometimes we are. So in this paper, the group identified ERG1C1, and that includes a membrane protein, and it's involved with how the protein is transported between what's called the endoplasmic reticulum, where protein synthesis takes place, and the Golgi apparatus, where the protein is modified. And we know now mutations can affect how genes are made and how they are then modified after they're made in either the endoplasmic reticulum or the Golgi. So in this slide here, this is a diagnostic rate using whole exome analysis. This is in the Forge Canadian Consortium here, and you can see you can get as high as 40% diagnostic yields. And if we're looking at, say, neuromuscular, 23% diagnostic yields, which for genetic testing is actually very high and very good. It's still not 100%, but it's certainly very hopeful for families as well. And this kind of justifies use in the clinic in terms of recommending this as a test. I have to qualify that every genetic testing, for the most part, does require insurance pre-authorization. So it's not just a box that I check and say order whole exome. It's a whole process. A genetic counselor has to spend time reviewing this and what the consequences of obtaining and not obtaining a result are. There can be other genes that people may or may not want to find out that are not necessarily related to the child's phenotype, but are important. For instance, hereditary breast and ovarian cancer, cardiomyopathies, other conditions that may affect blood vessels, large blood vessels, cancer-causing conditions as well. So there are other conditions that need to be explained to families that may come up that may not have impact now, but may have impact later on as well. So this is a child with distal arthrogryposis type 1, so you can see. And there are distal arthrogryposis next generation sequence panels as well. So here we're looking at one pathway, and we're testing genes in this particular pathway that are associated with distal arthrogryposis. So we may say, okay, this child looks like they have a distal arthrogryposis, and let's do this type of a gene panel, which is commercially available. So you've heard exomes. There's also whole genome. Whole genome is not available commercially. It is being used as a research tool. And in whole genome, we are looking at the entire genome. Whole exome, we're just looking at the coding portions of our genome as well. And 1% of the genome for a whole exome. Whole genome, we look at everything. We also look at copy number variants and what's called single nucleotide variants. So with exomes, we'll end up with 20,000 variants. So I don't see all of them. The lab kind of filters them out through a bioinformatic process. Whole genome, they'll be creating a million variants. So exome analysis is for what we call Mendelian. Mendelian is P. So traits that are caused by one gene passed down from parent to child, or parents may carry a gene, and the gene may only be significant clinically when there are two altered genes, or we call that recessive inheritance, as opposed to dominant inheritance, where a change in one gene is required for a condition to occur. Whole genome is more suitable for more complex types of genetic trait identification as well. And as I mentioned, we're not really using whole genome analysis in the clinic these days. So also, if you're trying to say, well, where's a good place to start? Generally speaking, I think for many conditions, especially arthrogryposis associated with birth defects, we like to start with a chromosome microarray as a first step. If that's normal, then we would think about other types of panel versus whole exome analysis as well. So, when we talked about Sanger analysis for one gene, we're targeting or testing to one or two types of genes, and we expect this to be very less heterogeneous, and we're looking at specific phenotype. So examples include conditions like cystic fibrosis, for instance, spinal muscular atrophy. Those are very specific phenotypes. But say if we've done some targeting, or even if we have a series of possibilities that could be part of one pathway, like distal arthrogryposis, then we would use a multi-gene panel. We would go more for a non-specific, less specific, more heterogeneous type of testing. If we have a non-specific phenotype, then we would consider whole exome or whole genome as a test of choice to go. And there's no wrongness in, say, starting off with a panel and then going to exomes after that as well. So we try to really use the information to solve the puzzle. We use the components of history, family history, physical exam, x-rays, lab testing, literature searching to solve the puzzle of identifying a syndrome. As Dr. Hall mentioned, the more specific cause is really the best that we can really hope to aim for in our patients, and that's what our goal is. Genetic counseling really is this communication process that aims to increase the family understanding of the genetic condition, discuss options regarding disease management and risks and benefits of further testings and options, and help the individual to identify the psychosocial tools that are required to cope with potential outcomes and reduce the family's anxiety. So genetic counselors are really trained to offer short-term psychotherapy and support for parents that have a child with a particular birth defect or genetic condition. All testing has some degree of insurance issue. Pretty much all the tests that we do require some insurance pre-authorization by and large. And insurance companies also these days would like to know how will genetic testing alter case management. And I also think for arthrogryposis, as more and more discoveries are being made, there's certainly that greater chance that testing will help with identifying therapies in the future. But even now, I advocate for genetic testing with everyone that I see because I think it's really important, and this does have an impact on parents, on the child's therapy as well. So we try to synthesize all the components of the genetic evaluation and provide a clear explanation as to whether or not the child has a condition with a genetic cause. This process will provide and does provide a more precise risk for future pregnancies in many instances. And some forms of arthrogryposis we know are not necessarily genetic, such as amyloidosis congenita. All genetic testing requires a degree of follow-up with the family and meeting with them to discuss these results. So genetics does keep getting more and more complex as time goes on. It's not as simple as we thought back 40 or so years ago. However, I certainly believe that there, based on what Dr. Hall mentioned, there's certainly future achievements are in store, and I think there can be nothing but hope and confidence as we approach the future. So thank you very much. Any questions?
How often do you suggest we go and seek genetic counseling and such? We had it done when she was 2 weeks old, and she's 8 now, and like Dr. Hall said, a lot of stuff happens between that amount of time.
Yeah, I think it would be a good idea to make an appointment. Certainly 6 years is a lot has changed in 5 years in terms of genetic testing, really. So,
we had a very definite finding because she had Sheldon Hall, so we know what that was. But even with that, there's still a lot of stuff going on.
Right, so if you have a known diagnosis, then certainly checking in with genetics and saying, are there any new treatments, therapies, or is my child getting everything that, has there been anything new in natural history for this condition? So some of my patients, if they have a known diagnosis, I'll say come back each year or 2 years as well, and we can go in the literature and say, this has been checked off the list to do, or maybe we should go back again, and hearing, vision, and other things, and so
Thanks.
For a family that their child came to them through adoption, so we have no family history, is genetics still a thing to explore even if we don't know that piece of the puzzle?
Yeah, no, absolutely. Now, I don't know what testing was done, but I talked about exome. We can still offer exomes with just what we call the proband or the index case, even if we don't have parents. Certainly, the more information we have, the better. And in my practice, I know sometimes the biologic parents might be willing to provide a DNA sample, but if there's no contact at all, that still doesn't eliminate the possibility of not doing what we call an exome. But that being said, the more information you have, the better it is. Thank you.